Bismillahir Rahmanir Rahim. LL37, the only member of the human cathelicidin family which has antiviral, antifungal, and antibacterial properties, its complexation with various nucleic acids has been shown to form diverse structures having immunostimulatory effects and contribute to numerous beneficial health outcomes, while on the other side, it can also cause harmful effects. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University, and here is our second video about LL37's complex dance with nucleic acids. First, let's start with LL37 and neutrophil extracellular traps. Neutrophils respond to inflammation by migrating to the site of infection and releasing web-like structures called the neutrophil extracellular traps or nets. These traps, composed of decondensed nuclear DNA and mitochondrial DNA bound to histones, granule proteins, and LL37, effectively capture and eliminate pathogens such as bacteria. Extracellular traps are not only produced by neutrophils to aid in microbial defense, but also by mast cells. Both cell types play key roles in the innate immune response, with mast cells known to initiate neutrophil recruitment to wound sites. Mast cell-derived extracellular traps resemble neutrophil extracellular traps and consist of web-like DNA strands with histones and antimicrobial peptides. The bacterial killing mechanism of antimicrobial peptides has traditionally been linked to their effects on membranes. However, recent studies suggest that the ability of antimicrobial peptides, particularly LL37, to permeabilize bacterial membranes and bind intracellular components such as nucleic acids or ribosomes plays a key role in their antimicrobial activity. Following the process of netosis, during which neutrophils release neutrophil extracellular traps after plasma rupture, these structures can be cleared by DNase 1 or possibly by macrophages. Effective clearance by neutrophil extracellular traps is crucial to avoid harmful effects associated with their persistence. Failure to efficiently remove nets has been linked to the progression of various diseases such as cancer, chronic rhinosinusitis, systemic lupus erythematosus, and COVID-19. Nets also may be responsible for the development of atherosclerosis and thrombosis. One antimicrobial peptide present in nets, LL37, significantly enhances the resistance of nets to degradation by Staphylococcus aureus nucleases. This protective effect could be attributed to the cationic nature of LL37, which binds to neutrophil DNA and shields it from nuclease-mediated degradation. It was discovered that LL37 DNA complexes are taken up and processed within macrophages, leading to the release of LL37, which targets and eliminates intracellular mycobacteria. This highlights the role of LL37 DNA complexes in supporting human defense mechanisms against intracellular bacterial infections in macrophages. In one study, a significant role of RNA rather than DNA within nets was identified in the context of psoriasis. This study demonstrated that self-propagating net and cytokine responses occurred independently of DNA. Instead, LL37 and RNA complexes activated toll-like receptor 8 and toll-like receptor 13 mediated cytokine production and net release both in vitro and in vivo, intensifying inflammation and driving a vicious cycle, thus sustain chronic inflammation. The formation of LL37 within biomacromolecular complexes has been identified as a key factor in the development of other autoimmune diseases. Specifically, dysregulation of LL37 is thought to contribute to systemic lupus erythematosus, with type 1 interferon pathways being implicated as major drivers of disease progression and severity. Neutrophils in systemic lupus erythematosus have been observed to undergo netosis, with LL37 proposed as a mechanism to stabilize DNA immunostimulatory molecule complexes rather than clear them in SLE patients. Complexes formed by DNA from netosis and LL37 and anti-double strand DNA antibodies activate toll-like receptor 9 in plasmacytoid dendritic cells, leading to increased interferon 1 production. Also, systemic LA patients showed autoantibodies against LL37, which promoted additional net formation. Together, these processes fuel an inflammatory cycle by generating more nets and further amplifying type 1 interferon production. The production of type 1 interferon by plasmacytoid dendritic cells in response to LL37 DNA complexes within nets has also been linked to atherosclerosis. A study showed that LL37 DNA complexes can stimulate plasmacytoid dendritic cells to produce interferon alpha in atherosclerotic arteries, leading to the overexpression of LL37 and enhanced net formation. Now let's talk about LL37 and RNA. RNA, like DNA, has a negatively charged backbone due to phosphate groups, which makes it an ideal binding partner for LL37. Similarly, LL37 can transport RNA into various types of cells. One study demonstrated that cathelicidin facilitates the intracellular uptake of single-strand RNA oligonucleotides. Also, LL37 has been shown to transport double-strand RNA into cells, triggering the expression of growth factors in keratinocytes and endothelial cells. Another study reported that LL37 and double-strand RNA work synergistically to upregulate toll-like receptor 3 in bronchial epithelial cells, enhancing the amount of imported double-strand RNA and activating downstream toll-like receptor 3 signaling. LL37 also has been shown to promote inflammation by facilitating the binding of self-RNA to cell surface scavenger receptors. LL34, a modified form of LL37, inhibit type 1 interferon expression when RNA was presented to keratinocytes and led to a reduction in inflammatory activity in keratinocytes as well as in epithelial and endothelial cells. A common naturally occurring post-translational modification of LL37 is called citrullination, in which peptidyl arginine residues undergo enzymatic deamination, converting the amino acid arginine into citrulline. This process is catalyzed by what's called PAD enzymes or peptidyl arginine deaminases, in which calcium acting as a key regulator of PAD activity. Citrullination of LL37 can result in the loss of positive charges and alterations to its alpha helical structure, thereby disrupting its molecular interactions with other biomolecules. Since the LL37 function relies on its cationic charges and alpha helical structure, this modification diminishes its anti-inflammatory and bactericidal properties. Citrullination of LL37 can occur during inflammatory responses, so caution is advised when designing drugs based on LL37, as they may exacerbate the severity of sepsis. Finally, as a summary, complexes formed by LL37 and various nucleic acids are potent immunomodulators that can profoundly affect the human innate immune system. LL37 functions as a nucleic acid condensing agent capable of creating diverse complex structures. The presence of other biomolecules can affect the complexation and condensation mechanisms of LL37 with nucleic acids, as different peptides assemblies may disrupt the electrostatic and aromatic interactions, which are the primary mechanisms driving the complexation of LL37 with nucleic acids. The dysregulation of LL37 has been implicated in various autoimmune diseases. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.